Hello, my name is Walker Liu, and today I will be doing my scientific research project. And my topic is the effects of bifidobacterium on gut microbiota, serotonin levels, stress, and anxiety on Faxonius virulis model organisms. So just a little bit of an introduction and background. Two key terms that are really important to truly understand my project are gut microbiota and the gut-brain axis. Gut microbiota is a complex and dynamic population of microorganisms that include bacteria, archaea, fungi that are located in the GI or the gastrointestinal tract. Although many different factors affect the human gut microbiota, diet is one of the main effectors of gut microbiota growth, specifically in the infant stage, but also in the short and long-term growth of an organism. So originally, research showed that mood, memory, and cognition were regulated exclusively by the central nervous system. However, with further research, it is becoming more clear that there are many non-nervous system factors, such as the GI tract, that plays a major role in our cognitive function related to microstructure and morphology. And that's what leads to gut-brain axis. The gut-brain axis is the two-way biochemical signaling that takes place between the GI tract and the central nervous system. And you can kind of see it here with this little picture right here to the left. As you can see, the brain and the gut microbiota are intertwining and creating biochemical signals that relate to each other. So given this information, researchers are then presented with the question on how diet affects the gut microbiota and the GI tract, and how the change in gut microbiota affects the central nervous system. So I will be using crayfish, or more specifically the Faxonius virulis species, as my model organism for this project. And there's a slight difference between crayfish and humans when it comes to the release of hormones or serotonin receptors. So crustacean hypoglemic hormone, or CHH, is a neuropeptide that is synthesized, stored, and released by brain and eye stalk structures in decapods such as crayfish. Hypoglycemia is a common stress response of many aquatic animals. In vertebrates, however, hypoglycemia is mediated by elevated plasma cortisol, whereas in crustaceans, it occurs following the involvement of CHH produced in the eye stalk. So overall, the purpose of this study is to analyze the effects of bifidobacterium on gut microbiota, stress, and anxiety of crayfish. Bifidobacterium suppresses anxiety-like behaviors, restoring non-adrenaline levels, including GABA production or protecting gut dysbiosis. Supplementation with bifidobacterium may prevent the onset of depression from stress in humans. In addition, supplementation of bifidobacterium may present or minimize relapse from remission induced by inflation and/or stress in depressed patients. Based on these past studies, the hypothesis is that an increase in bifidobacterium in the diets of crayfish will decrease stress and anxiety. This will be shown through behavior tests along with qPCR. So if you move on to my methods, my method is I have 4 different crayfish tanks, and each crayfish tank has 4 crayfish. So there's a control tank, and then there's a variable tank. Each variable tank will be fed a varying amount of bifidobacterium, 1%, 2%, and 5%. And they'll be fed twice a week for 4 weeks. One thing that is really important to learn about my method section is the creation of the food. So because bifidobacterium is a very water-soluble food, or very water-soluble bacteria, you can't simply just put the bacteria in the water, it will just dissolve. So what I had to do is I had to take the control food, crush it up, place the bacteria in the food, mash it all together, and recreate the food again, so it's easier for these crayfish to consume. And then I will be doing qPCR RNA extraction and looking at the 5-HT1A receptor, also known as the serotonin receptor. So my results have yet to be collected. My expected date is February 24th. Due to the fact that I have been feeding these crayfish for 3 weeks now, almost a month, is that it is taking a little long to get the results. But what I'm looking for in the results is I will be doing qPCR and I will be doing RNA extraction. And like I said earlier, I will be looking at that 5-HT1A receptor, also known as the serotonin receptor. Discussion. So predicted results. So this is just my predicted results. Obviously, I don't have any results yet, but this is what I hope to happen, and this is what I plan on happening. Is I plan that as bifidobacterium increases through the variable diets, so from 1% to 2% to 5%, the qPCR will show an increase in 5-HT1A receptors, or the serotonin receptors, which means an increase in serotonin levels, which then in turn means a decrease in stress and anxiety, which is what I'm looking at. Conclusion, so just the bigger picture. So what does this truly mean if the hypothesis turns out correct? So bifidobacterium could then be used as a probiotic. It is used in some instances. Small niche groups of people take bifidobacterium supplements like you would take a vitamin C or vitamin D supplement. But if this research And if the hypothesis stands, it could really make bifidobacterium a mainstream probiotic, and just something you take every single day during your breakfast or once or twice a day. It's just a daily like pill or something like that. And in addition, it could also be used as a substitute for antidepressants. Antidepressants are really hard to get. They're very expensive and they require prescriptions. And this would just make it a lot easier. And you could just get it at the store. You could get it at a Target or a Costco or something like that. It just make it really easy to get antidepressants for people in need. Here are my references. So here are just a few of the references that I use a lot during my project. And that is the conclusion of my project. I hope you learned a lot, and I'm looking forward to hearing your responses.